Welcome to the Pharmaceutical Technology Podcast, PDA Technical Report 83, Virus Contamination in Biomanufacturing, Risk Mitigation, Preparedness, and Response. This podcast is brought to you by PDA, the leading provider of science, technology, regulatory information, and education for the biopharmaceutical community. To find out more, please visit them on the web at www.pda.org. And now, here's your host for this podcast, the contributing editor for Pharmaceutical Technology, Cindy Dubin.
Hello, everyone. This is Cindy Dubin, contributing editor for Pharmaceutical Technology, and I'm here with Dr. Robert D. Kiss, Vice President of Process and Analytical Development at Sutro Biopharma, who has spent more than 25 years developing and supporting mammalian cell culture processes used for the production of biopharmaceuticals. Thank you for being here today, Bob.
Thank you very much for having me. I appreciate this opportunity.
Sure. So to get started, what is the purpose of this technical report?
Well, this technical report was generated to provide guidance regarding best practices and proven effective means of preventing virus contaminations in biopharmaceutical production processes derived from mammalian cell cultures. In addition, the guidance here is provided in how to prepare for a contamination event and how to respond should one occur. This should be particularly valuable for companies who have not yet experienced a significant virus contamination event. You'll note that I use the term yet experienced. While virus contaminations in biopharmaceutical processes are thankfully very rare events, they are extremely impactful to the company affected, as well as potentially to the entire industry, depending on the circumstances. Having been involved in the response to a significant virus contamination in a biologics manufacturing facility, I know how impactful this can be to an organization and potentially to the supply of products to patients that need them. As I mentioned, thankfully, biopharmaceutical production has had a very successful history in terms of product safety with respect to this type of virus contamination. That's in part due to the inherent low probability that is a result of the extensive testing of the cells that are used in the processes before they're used, after they're used, control of raw materials that are used in the processes, and because the industry has developed validated virus clearance steps to protect the products. However, the combination of this low probability of occurrence with a very high severity of the impact makes for a challenging situation from a risk management perspective. Most in the industry have acknowledged that the best way to address this is through applying appropriate detection, response, and remediation planning. We want to keep this type of event at bay in being an if situation and not ultimately manifesting itself into a when scenario.
So, based on your last statement, is that why Parenteral Drug Association and the author team feel that this is an important topic to be addressed through a PDA technical report?
Exactly. The biotech and pharma industries often look to PDA technical reports for these types of consolidated summaries of experiences, best practices, and approaches for managing and interacting with health authority guidelines. The value of PDA technical reports is really quite clear for practitioners within these industries. And the PDA is also an excellent venue under which to assemble experts from across multiple sources who come together and write these types of important guidances, as was the case with this technical report, where more than half the author team has had personal experiences dealing with virus contaminations in production scale cell culture processes.
And Bob, who does the author team believe should read this technical report?
Well, we feel that this technical report will be of value to biopharmaceutical professionals involved all along the supply chain in the production of biologic products derived from mammalian cell culture processes. So, for example, this could include anyone involved in supporting an upstream cell culture process, from the facility and equipment design side of things to procuring raw materials to the process development folks who develop the production process, to the manufacturing operations group that executes the process, to the quality control group that tests and performs virus screening, for example, to the quality assurance staff that are overseeing the production, to the compliance staff that are ensuring that the company is complying with applicable regulations, and even to staff and regulatory affairs. It really does cut across the entire supply chain, and we've tried to address all those aspects in this document. We cover many aspects of preventing, preparing for, and importantly, responding to a virus contamination in a mammalian cell culture process should it occur. And this includes how to prepare from a risk assessment, risk management perspective, from a testing perspective, from a virus identification standpoint, how to decontaminate process equipment and facilities, how to prepare and govern the process of returning a facility to service after a contamination event, which is really an important part of managing a situation should one occur. And finally, communications, both within your organization and with health authorities should an event of this type occur. And in addition, I wanted to mention that the information presented in this technical report, we consider it also relevant to some of the emerging production processes for gene and cell therapies, also known as advanced therapy medicinal products, or ATMPs. So we're hoping that the audience will include folks from really all facets of biologics production.
Definitely a lot of information packed into this. So could there possibly be anything else you'd like to say about the tech report and its relevance to the biopharmaceutical industry?
Well, thanks for asking that. Really, a lot of work has been done over the last, I'd say, 8 or so years on this topic by an existing industry consortium. It's the Consortium on Adventitious Agent Contamination in Biomanufacturing. It's known as CAACB. And I mention that because this group has conducted surveys of the industry to generate data on observed incidences of virus contamination in biopharmaceutical manufacturing and how companies have managed these rare but really very impactful events. And the consortium has presented some of those findings publicly and is planning to publish a manuscript in the very near future, which will share those survey findings so that the entire industry can also learn from the experience of others who have had these types of events occur. So that will be out there soon, and I think really will marry well to this technical report, which is intended to help people determine how best to prepare and how to address these types of situations should they occur.
Well, I hope you will keep us posted on those survey findings once they become available.
Happy to do so, and look forward to this technical report being available to folks in the PDA and more broadly, and this type of information in general, because product safety is such an important aspect of what we do in the industry.
Well, we appreciate your time today. Thank you so much for this informative overview, Bob.
You're welcome.
This has been Cindy Dubin, contributing editor for Pharmaceutical Technology. Thanks to all for listening.
You've been listening to the Pharmaceutical Technology Podcast, PDA Technical Report 83, Virus Contamination in Biomanufacturing, Risk Mitigation, Preparedness, and Response. This podcast is brought to you by PDA, a leading provider of science, technology, regulatory information, and education for the biopharmaceutical community. To find out more, please visit them on the web at www.pda.org.